Hello, smart scholars. This is Dr. Eve Mboso. Welcome to another session. Today, we are going to be looking at enzymes. In the previous section, we looked at metabolic pathways, and we said that metabolic reactions are catalyzed by enzymes. So what are enzymes? Enzymes are biocatalysts that speed up the rate of biochemical reactions. And they achieve this by lowering the activation energy. So now let's look at this energy reaction progress. Now this is the energy required to catalyze a biochemical reaction. This is an uncatalyzed biochemical reaction. Now what enzymes do is that they lower the activation energy so that the substrates are stabilized when the enzymes bind to the substrate and they progress faster and form products. Enzymes also have clinical implications. Enzymes are used to check the health status of individuals. Blood levels are checked for enzyme concentrations, and these are used to check health statuses and for other clinical assays. So now, what happens? Enzymes bind to the active site, of the substrate, and they form an enzyme substrate complex. When this happens, the activation energy is lowered, and this makes the substrate more stable, and then the reaction progresses faster and products are being formed. Now, there are 6 classes of enzymes based on the reactions they catalyze. These enzymes are put into these classes: oxidoreductases, transferases, okay, I'll use this marker, hydrolysis, you have lyases, isomerases, and ligases. So these are the 6 classes of enzymes. The oxidoreductases, they catalyze oxidation, reduction reactions. And then the transferases, they are amidotransferases, amino acid transferases. These reactions, this class of enzymes catalyze the transfer of an amino group or functional groups depending on the reaction. So they are involved in the transfer of functional groups from the substrate to form products. Then you have the hydrolysis. This class of enzymes, what they do is that they split molecules with the use or the addition of water. And then, unlike the transferases, these hydrolysis their reactions are mostly not reversible. Now, you have the lyases. The lyases are a class of enzymes, that are involved in splitting carbon-carbon, carbon-nitrogen atoms. Then you have the next class of enzymes, which are known as the isomerases. These enzymes are responsible for forming isomers. Now, what do? They move reactions or molecules from one point of a biomolecule to the other. For example, the formation of glucose and fructose, which are isomers, okay, you have it in the glycolytic pathway. They change from glucose 6-phosphate to fructose 6-phosphate. So the isomerase enzymes plays a major role in these class of reaction. Then you have the lyases. Now, this class of enzymes, they're involved in splitting or breaking or adding of substrate groups. But this class of enzymes make use of ATP. So you can also call these lyases synthetases because they require ATP for their reaction. Now, enzyme reactions must actually be regulated. So you have 2 major ways they can be regulated: by covalent modification, where functional groups are added to the protein to change the activity of the enzyme. So what happens is that you can have, like in post-translational modification, in the post-synthesis of RNA. Now, in the post-translational modification, something happens, functional groups are added to the protein, and this alters the activity of the enzyme. Then another way enzymes can be regulated is through allosteric modification. Now, the word allosteric is from the root word allo, which means another. So what happens is that these compounds bind to another site on the enzyme apart from the active site. Now what they do is that when they bind, they change the sensitivity of the enzyme, all right, to the substrate. In other words, they modulate the activity of the enzyme. While in covalent modification, there is a covalent bond that is being formed. So when this happens, all right, you can either have a positive modulation or a negative modulation where the enzyme, all right, becomes inactive. So that's all for now. Drop your comments in the comment section below and see you next time.